kingdom biology.
In biology, kingdom is the second highest taxonomic rank below domain. Kingdoms are divided into smaller groups called phyla. Traditionally, textbooks from the United States used a system of 6 kingdoms while British, Australian and Latin American textbooks used 5 kingdoms. Some recent classifications based on modern cladistics have explicitly abandoned the term kingdom, noting that the traditional kingdoms are not monophyletic, that is, do not consist of all the descendants of a common ancestor.
Definition and associated terms.
When Carl Linnaeus introduced the rank-based system of nomenclature into biology, the highest rank was given the name kingdom, and was followed by 4 other main or principal ranks, the class, order, and genus. Later 2 further main ranks were introduced, making the sequence kingdom, phylum or division, class, order, family, genus and species. In the 1960s a rank was introduced above kingdom, namely domain, or empire, so that kingdom is no longer the highest rank. Prefixes can be added so subkingdom and infrakingdom are the 2 ranks immediately below kingdom. Superkingdom may be considered as an equivalent of domain or empire or as an independent rank between kingdom and domain or subdomain. In some classification systems the additional rank branch, Latin, ramus, can be inserted between subkingdom and infrakingdom, for example Protostomia and Deuterostomia in the classification of Cavalier-Smith.
Modern view. The 3 domains of life.
From around the mid-1970s onwards, there was an increasing emphasis on comparisons of genes on the molecular level, initially ribosomal RNA genes, as the primary factor in classification. Genetic similarity was stressed over outward appearances and behavior. Taxonomic ranks, including kingdoms, were to be groups of organisms with a common ancestor, whether monophyletic, all descendants of a common ancestor, or paraphyletic, only some descendants of a common ancestor. Based on such RNA studies, Carl Oeys thought life could be divided into 3 large divisions and referred to them as the 3 primary kingdom model or a kingdom model. In 1990, the name domain was proposed for the highest rank. Oeys divided the prokaryotes, previously classified as the kingdom Monera, into 2 groups, called eubacteria and archaebacteria or archaea, stressing that there was as much genetic difference between these 2 groups as between either of them and all eukaryotes. According to genetic data, although eukaryote groups such as plants, fungi, and animals may look different, they are more closely related to each other than they are to either the eubacteria or archaea. It was also found that the eukaryotes are more closely related to the archaea than they are to the eubacteria. Although the primacy of the eubacteria-archaea divide has been questioned, it has been upheld by subsequent research. There is no consensus on how many kingdoms exist in the classification scheme proposed by Oeys.
How many kingdoms are present in the eukarya?
In 2004, a review article by Simpson and Roger noted that the protista were a grab bag for all eukaryotes that are not animals, plants or fungi. They held that only monophyletic groups should be accepted as formal ranks in a classification and that, while this approach had been impractical previously, necessitating literally dozens of eukaryotic kingdoms, it had now become possible to divide the eukaryotes into just a few major groups that are probably all monophyletic. On this basis, the diagram opposite, redrawn from their article, showed the real kingdoms, their quotation marks, of the eukaryotes. A classification which followed this approach was produced in 2005 for the International Society of Protistologists, by a committee which worked in collaboration with specialists from many societies. It divided the eukaryotes into the same 6 supergroups. The published classification deliberately did not use formal taxonomic ranks, including that of kingdom. In this system the multicellular animals, metazoa, are descended from the same ancestor as the unicellular choanoflagellates and the fungi which form the opisthokonta. Plants are thought to be more distantly related to animals and fungi. However, in the same year as the International Society of Protistologists classification was published, 2005, doubts were being expressed as to whether some of these supergroups were monophyletic, particularly the chromalvolata, and a review in 2006 noted the lack of evidence for several of the supposed 6 supergroups. As of 2010, there is widespread agreement that the Rhizoria belong with the Stramenophiles and the Alvolata, in a clade dubbed the SAR supergroup, so that Rhizoria is not one of the main eukaryote groups. Beyond this, there does not appear to be a consensus. Ragozin et al. In 2009 noted that the deep phylogeny of eukaryotes is an extremely difficult and controversial problem. As of December 2010, there appears to be a consensus that the 2005-6 supergroup model does not reflect the true phylogeny of the eukaryotes and hence how they should be classified, although there is no agreement as to the model which should replace it.
Historical development.
The classification of living things into animals and plants is an ancient one. Aristotle, 384-322 BC, classified animal species in his History of Animals, while his pupil Theophrastus, c. 371 c. 287 BC, wrote a parallel work, the Historia Plantrum, on plants. Carolus Linnaeus, 1707-1778, laid the foundations for modern biological nomenclature, now regulated by the nomenclature codes, in 1735. He distinguished 2 kingdoms of living things, regnum animale, animal kingdom, and regnum vegetable, vegetable kingdom, for plants. Linnaeus also included minerals in his classification system, placing them in a 3rd kingdom, regnum lapidum. In 1674, Antony van Leeuwenhoek, often called the father of microscopy, sent the Royal Society of London a copy of his first observations of microscopic single-celled organisms. Until then, the existence of such microscopic organisms was entirely unknown. Despite this, Linnaeus did not include any microscopic creatures in his original taxonomy. At first, microscopic organisms were classified within the animal and plant kingdoms. However, by the mid-19th century, it had become clear to many that the existing dichotomy of the plant and animal kingdoms rapidly blurred at its boundaries and outmoded. In 1866, Ernst Haeckel proposed a 3rd kingdom of life, the Protista, for neutral organisms, which were neither animal nor plant. Haeckel revised the content of this kingdom a number of times before settling on a division based on whether organisms were unicellular, Protista, or multicellular, animals and plants. The development of the electron microscope revealed important distinctions between those unicellular organisms whose cells do not have a distinct nucleus, prokaryotes, and those unicellular and multicellular organisms whose cells do have a distinct nucleus, eukaryotes. In 1938, Herbert F. Copeland proposed a 4-kingdom classification, elevating the protist classes of bacteria, Monera, and blue-green algae, Phycochromaceae, to phyla in the novel kingdom Monera. The importance of the distinction between prokaryotes and eukaryotes gradually became apparent. In the 1960s, Sternier and Van Niel popularized Edouard Chatton's much earlier proposal to recognize this division in a formal classification. This required the creation, for the first time, of a rank above kingdom, a superkingdom or empire, later called a domain. The differences between fungi and other organisms regarded as plants had long been recognized by some. Haeckel had moved the fungi out of Plantae into Protista after his original classification, but was largely ignored in this separation by scientists of his time. Robert Whittaker recognized an additional kingdom for the fungi. The resulting 5-kingdom system, proposed in 1969 by Whittaker, has become a popular standard and with some refinement is still used in many works and forms the basis for new multi-kingdom systems. It is based mainly upon differences in nutrition. His plantae were mostly multicellular autotrophs, his animalia multicellular heterotrophs, and his fungi multicellular saprotrophs. The remaining 2 kingdoms, Protista and Monera, included unicellular and simple cellular colonies. The 5-kingdom system may be combined with the 2-empire system. In the Whittaker system, plantae included some algae. In other systems, for example, Margulis system, plantae included just the land plants, Embryophyta. Despite the development from 2 kingdoms to 5 among most scientists, some authors as late as 1975 continued to employ a traditional 2-kingdom system of animals and plants, dividing the plant kingdom into subkingdoms: Prokaryota, bacteria and cyanophytes, Mycota, fungi and supposed relatives, and Chlorota, algae and land plants.
Other proposed systems. 8 kingdoms.
Thomas Cavalier-Smith thought at first, as it was nearly consensually admitted at that time, that the difference between eubacteria and archaebacteria was so great, particularly considering the genetic distance of ribosomal genes, that they needed to be separated in 2 different kingdoms, hence splitting the empire bacteria into 2 kingdoms. Eubacteria was divided into 2 subkingdoms, negabacteria, gram-negative bacteria, and Proteobacteria, gram-positive bacteria. Technological advances in electronical microscopy allowed the separation of the chromaster from the plantae kingdom. Indeed, the chloroplast of the chromasts is located in the lumen of the endoplasmic reticulum instead of in the cytosol. Moreover, only chromasts do contain chlorophyll C. Since then, many non-photosynthetic phyla of protists, thought to have secondarily lost their chloroplasts, were integrated into the kingdom chromaster. Finally, some protists lacking mitochondria were discovered. As mitochondria were known to be the result of the endosymbiosis of a proteobacterium, it was thought that these amitochondriate eukaryotes were primitively so, marking an important step in eukaryogenesis. As a result, these amitochondriate protists were separated from the protist kingdom, giving rise to the, at the same time, superkingdom and kingdom Archezoa. This was known as the Archezoa hypothesis. This superkingdom was opposed to the Metakaryotes superkingdom, grouping together the 5 other eukaryotic kingdoms: Animalia, Protozoa, fungi, plantae and chromaster.
6 kingdoms.
In 1998, Cavalier-Smith published a 6-kingdom model, which has been revised in subsequent papers. The version published in 2009 is shown below. Compared to the version he published in 2004, the alveolates and the rhizarians have been moved from kingdom Protozoa to kingdom chromaster. Cavalier-Smith no longer accepts the importance of the fundamental eubacteria-archaebacteria divide put forward by Oes and others and supported by recent research. His kingdom bacteria includes archaebacteria as a phylum of the subkingdom monobacteria, which comprises only one other phylum, the Proteobacteria. The 2 subkingdoms Anabacteria and Negabacteria of kingdom Bacteria, sole kingdom of empire Prokaryota, are opposed according to their membrane topologies. The bimembranous-unimembranous transition is thought to be far more fundamental than the long branch of genetic distance of archaebacteria, viewed as having no particular biological significance. Cavalier-Smith does not accept the requirement for taxa to be monophyletic, holophyletic in his terminology, to be valid. He defines Prokaryota, bacteria, negabacteria, Anabacteria and Proteobacteria as valid paraphyletic, therefore monophyletic in the sense he uses this term, taxa, marking important innovations of biological significance, in regard of the concept of biological niche. In the same way, his paraphyletic kingdom Protozoa includes the ancestors of Animalia, fungi, plantae and chromaster. The advances of phylogenetic studies allowed to realize that all the phyla thought to be archezones, that is primitively a mitochondriate eukaryotes, had in fact secondarily lost their mitochondria, most of the time by transforming them into new organelles, hydrogenosomes. This means that all living eukaryotes are in fact metakaryotes, according to the significance of the term given by Cavalier-Smith. Some of the members of the defunct kingdom Archezoa, like the phylum Microsporidia, were reclassified into kingdom fungi. Others were reclassified in kingdom Protozoa like Metamonida, which is now part of infrakingdom Excavata. The diagram below does not represent an evolutionary tree.
Viruses.
There is ongoing debate as to whether viruses, obligate intracellular parasites that are not capable of replication outside of a host, can be included in the tree of life. A principal reason for inclusion comes from the discovery of unusually large and complex viruses, such as mimivirus, that possess typical cellular genes.
Summary.
A summary of the different kinds of proposed classification schemes presented in this article is summarized in the table below. The kingdom-level classification of life is still widely employed as a useful way of grouping organisms. There is no current consensus on how many kingdoms are present in the eukarya. In 2009, Andrew Roger and Alistair Simpson emphasized the need for diligence in analyzing new discoveries. With the current pace of change in our understanding of the eukaryote tree of life, we should proceed